Imagine your cell is like this massive, relentlessly busy Amazon fulfillment center.
Right.
Millions of proteins are the packages, constantly moving around on these complex, microscopic conveyor belts. Now imagine a system designed to slap a highly specific VIP shipping label onto select packages.
A label that dictates absolute fate.
Exactly. It determines whether a protein gets immediately activated, sent straight to the cellular incinerator, or granted all-access VIP clearance to the executive boardroom, which, in a cell, is the nucleus.
And in a healthy cell, that labeling system is, well, it's the ultimate organizational tool. It keeps the warehouse running smoothly, degrading damaged goods, and promoting healthy growth.
But what we're unpacking today is what happens when a rogue manager takes over that labeling machine.
Total chaos.
Which is exactly the focus of this deep dive. Welcome in. We're tearing into this massive 2026 review article from Springer Nature's Cell Death and Disease.
It's titled Decoding Neddylation in Malignancies.
Right. So we're going to completely deconstruct this cellular labeling mechanism called neddylation. We'll explore the terrifying ways cancer, hijacks this system to fuel its own growth, blind our immune system, and, this is the crazy part, effortlessly shrug off our most advanced chemotherapy drugs.
And more importantly, we're looking at the cutting-edge chemical warfare scientists are using to jam the gears of that rogue machine.
So have you ever wondered how cancer cells manage to outsmart our most advanced chemo drugs? Well, the answer might just lie in these tiny chemical post-it notes.
It really is a paradigm-shifting piece of literature, mostly because it forces us to look at cancer not just as a genetic mutation, but as an incredibly sophisticated warehouse logistics hack.
Right, a logistics hack. And the entire foundation of this hack rests on the label itself, this tiny protein known as NEDD8. But I want to visualize this label before we get into how it breaks. What exactly is a NEDD8?
So NEDD8 is incredibly small. I mean, it's an 81 amino acid, ubiquitin-like polypeptide.
Ubiquitin-like, meaning it's a tag.
Exactly. In the grand scheme of cellular proteins, it is basically a rounding error in size. It weighs in at just 9 kilodaltons.
Wow, 9.
Yeah, tiny. But its sole biological purpose is profound. It's meant to be covalently, meaning permanently and powerfully attached to a specific docking site on a target protein.
And that docking site is usually a specific amino acid, right?
Yes, almost always an amino acid called lysine.
Okay, but the cell doesn't just casually toss these labels around. Sticking with that fulfillment center analogy, the cell uses a highly restricted, energy-intensive assembly line to get that label onto a package.
It's a strict relay race. A multi-step conveyor belt.
Walk us through the workers on this line, because we need to understand the normal machine before we break it.
Well, before the label even hits the conveyor belt, it has to be prepped. Think of the NEDD8 precursor as a sticker that still has its protective backing on.
Oh, I like that.
Right, so in a phase called maturation, specific hydrolase enzymes, specifically UCHL3 and NEDP1, act like molecular scissors. They snip off the tail end of the NEDD8 protein.
Killing off the backing.
Exactly. And doing that exposes a highly reactive glycine or glycine chemical motif. Without peeling off that backing, the sticker can't adhere to anything.
Okay, so the backing is peeled off, the sticky glycolase side is exposed. Who's the first worker on the line to grab it?
That would be the E1 activating enzyme. You can picture E1 as the heavy lifting intake worker. In human cells, this E1 is a heterodimer made of APP, BP1, and UBA3.
Okay, so E1 grabs the label.
Right. E1 grabs the NEDD8 label, but to make it truly ready for attachment, it has to burn cellular energy, ATP.
Got it.
By burning ATP, the E1 worker forges a high-energy thioester bond with the label. It's like winding up a heavy spring-loaded trap.
So the label is locked in, just humming with potential energy.
Exactly. E1 hoists it up, ready to pass it down the line.
That makes sense. The label is energized, but E1 isn't the one applying it to the final package, right?
No, it has to hand it off. E1 passes that spring-loaded label to the second worker, the E2 conjugating enzyme like UBE2M or UBE2F.
Okay, E2 takes the baton.
Yep. This handoff happens through a seamless chemical swap. Now, the E2 worker is holding the energized label, carrying down the conveyor belt. But there's a catch.
What's the catch?
The E2 worker is essentially blind. It's carrying the VIP label, but it has absolutely no idea which of the millions of protein packages passing by actually deserves it.
Oh, wow. So if E2 is blind, there has to be a barcode scanner or something.
Exactly. Someone who recognizes a specific package and tells E2 where to put it.
And that would be the E3 ligase.
You got it. The E3 ligase is the barcode scanner. It's the third and most crucial worker on the line. E3 ligases like RBX1 or MDM2 act as the matchmakers.
So they grab the E2 worker and the package at the same time.
Precisely. The E3 physically grabs the E2 worker holding the label, simultaneously scans and grabs the correct target protein, aligns them perfectly, and triggers the final reaction.
Bam. The label is officially slapped onto the target's lysine docking site.
Fate sealed. And they target huge regulatory complexes. For instance, cullins, which are these scaffold proteins that control about 20% of all protein degradation in the cell.
20%.
Yeah, and non-cullins too, like the famous tumor suppressor P53.
Okay, but if the cell is constantly labeling things, doesn't it run out of labels? I mean, is there a way to undo this?
Oh, it is entirely reversible. That's a key part of the quality control process. The cell employs denatylases enzymes, like the COP9 signalism.
So they're like the quality assurance team at the end of the line.
Right. If a package doesn't need its VIP status anymore, this team chemically snips the NEDD8 label right back off, returning the protein to its original state and recycling the label for the next shift.
Okay, let me pause here to make sure I'm picturing this whole warehouse algorithm clearly, because this really is the foundation of everything cancer manipulates.
It is.
So we have a tiny VIP label called NED8. It gets prepped, then handed down an assembly line. E1 uses energy to hoist it. E2 blindly carries it. E3 scans the package and slaps the label on. And this label completely dictates the protein's destiny.
Usually sending it to cullins to be destroyed or radically changing how the protein behaves.
Exactly. So to you listening, now that you can visualize this highly controlled, beautifully balanced relay race, what do you think happens when a cancer cell floods the track with extra runners?
Well, absolute chaos.
Right. But how does a microscopic sticker physically alter a massive protein enough to cause a disease like cancer?
To understand the chaos, we really have to look at the physics of the proteins themselves. When you slap a NEDD8 label onto a protein, it isn't just a passive tracking number. It exerts physical force.
It changes the shape.
Dramatically. It radically changes the protein's three-dimensional shape, and by extension, its access to restricted areas of the cell.
Give me an example of this shape shifting. Like, how does a label physically bend a protein?
Let's look at triple-negative breast cancer, which is notoriously aggressive. In this cancer, the rogue warehouse manager starts overlabeling a specific E3 ligase, called TIM25.
Okay, TIM25.
Normally, TIM25 is folded up in a way that creates steric hindrance.
Okay, steric hindrance is a bit dense for anyone who hasn't taken organic chemistry in a while.
Fair enough.
The way I visualize it, it's like a factory robotic arm that's heavily wrapped in like thick, rigid winter coats or shrink wrap. It's too bulky. It just can't bend its joints to reach out and grab the materials it needs to work on.
That's a brilliant way to put it. So when the Neddylation assembly line slaps an NEDD8 label onto a highly specific spot on TIM25, specifically at lysine 117, the chemical bulk of that label acts like a blade cutting the shrink wrap.
It takes the bulky coat off.
Exactly. It forces the protein to physically unfold. Suddenly, the steric hindrance is gone. The robotic arm can swing freely.
And what does it do once it's free?
Because of this new shape, TRM25 becomes hyperactive. It aggressively starts turning on survival and recycling genes. As a direct result, those breast cancer cells develop a fierce structural resistance to standard chemotherapy drugs.
Like paclitaxel.
Yes, paclitaxel resistance specifically.
That is wild. The drug isn't failing because it's a bad drug. It's failing because the cancer cell took the shrink wrap off its own survival machinery.
Precisely.
And it doesn't stop it just unfolding things. In the source material, there's this concept of the label acting as an all-access backstage pass.
Yes, the relocation trick.
Okay.
Let's take PTN, which is, well, it's one of the human body's most famous tumor suppressors.
Like a security guard.
Right, a security guard that normally patrols the main floor of the cell, the cytoplasm, keeping growth in check. But in certain cancers, the assembly line forces a NEDD8 label onto PTN at specific residues, K197 and K402.
And this label acts as a molecular passport.
Exactly. It gives it VIP club access. It physically imports PTN out of the cytoplasm and straight into the nucleus,
Yeah.
the executive boardroom.
So it locks the security guard in the boardroom, but does it just trap it there or does it do damage?
Oh, it's worse than just trapping it. Once inside the nucleus, this modified PTN actually switches allegiances. It stops suppressing tumors and starts aggressively stimulating the production of fatty acids, which the tumor uses as raw building blocks to grow larger.
Unbelievable. Okay, if the tumor is growing that rapidly, it needs massive amounts of energy. This brings us to a classic hallmark of cancer, the Warburg effect.
Right, metabolic rewiring.
Cancer cells are basically addicted to sugar. They binge on glucose and ferment it, even when they have plenty of oxygen. How does this tiny VIP label drive that metabolic rewiring?
So the Warburg effect creates a highly acidic, sugar-rich environment. Now, you would think this toxic environment would trigger the cell's internal alarms.
Like P53.
Exactly. The master protein, P53, normally acts as the ultimate emergency brake. If a cell is acting erratically, P53 forces it to stop dividing or just self-destruct.
So why doesn't P53 hit the brakes when the cell starts binge-eating sugar?
Because the rogue manager uses the Neddylation line to destroy the brakes. The high glucose environment physically forces the quality assurance team, the denetylases, to walk away from their stations.
The copy 9 guys.
Right. Without quality control, a massive garbage disposal complex called CRL4 becomes permanently labeled and hyperactive.
It's just left in the on position.
Yes, and this hyperactive garbage truck has one primary target, wild-type P53. It hunts down the P53 brakes, chews them up, and allows the cancer to consume sugar and divide without any internal oversight.
Wait, let me push back on this for a second. If the cancer cell is binging on sugar, mutating its own security guards, and running this labeling machine at 1,000,000 miles an hour, I mean, it's making a massive mess.
Oh, a huge mess.
So why doesn't the immune system notice? Macrophages are the immune system's roving garbage trucks. They should be recognizing these rogue cells and swallowing them whole.
That is the exact logical assumption.
Right?
But cancer uses this same labeling system to effectively turn invisible.
Invisible? How?
Well, macrophages are powerful, but they operate largely by tactile feedback. They feel the surface of other cells looking for specific flags. One of those flags is an integrin on the surface of the tumor cell that basically says, I am damaged, eat me.
So the tumor pulls the flag down.
Precisely. And it does it by manipulating the labels. In colon cancer, for example, the tumor cell intentionally removes the NEDD8 label from an internal signaling protein called SHP2.
They denetylate it.
Right, specifically at K358 and K364. By taking that specific label off, it triggers a chain reaction that physically retracts the eat me integrin from the surface of the cell.
Wow.
The macrophage bumps right into the colon cancer cell, feels a smooth, normal surface without the flag, and just swims right past it. The tumor hides in plain sight.
It completely blinds the immune system. And if that isn't enough, the review notes it also destroys the cell's suicide switch.
Division of apoptosis, yeah. A healthy cell exposed to heavy radiation is programmed to undergo apoptosis, clean, organized cell death.
And a protein called NOXA is the trigger for this suicide sequence, right?
Yes. But in lung cancer, the UBE2FE2 conveyor belt worker becomes hyperactivated. It constantly churns out labels that direct NRXA straight to the cellular incinerator.
So the cell loses its ability to die.
It essentially becomes biologically immortal, shrugging off lethal doses of radiation because the NOXA trigger is just gone.
Okay, so let's quickly synthesize everything we just covered at this 10-minute mark.
Big idea.
This rogue Neddylation assembly line unfolds constrained robotic arms to fight off chemo. It hands out backstage passes to mutate security guards. It permanently destroys the cell's emergency brakes to feed its sugar addiction, and it pulls down the eat me flags to become totally invisible to our immune system.
It's a comprehensive survival strategy.
It really is. But here's the million-dollar question for you listening. An Amazon warehouse doesn't just go into overdrive for no reason.
Right.
Someone has to give the order. What is the outside pressure, the trigger, that forces the cancer cell to hit the print label button so aggressively in the first place?
The biological reality is that cancer is the ultimate opportunistic scavenger. The Neddylation machine is exquisitely sensitive to cellular stress. When a cell is stressed, its instinct is to adapt and survive. Cancer just exploits that survival reflex.
So it's not just a genetic accident. Let's break down these stresses, starting with viral hijacking. How does a virus walk into this warehouse and take over the assembly line?
A textbook example is the hepatitis B virus, which is a massive global driver of liver cancer.
HBV.
Right. When Hep B infects a liver cell, it doesn't just quietly replicate. It produces a specialized viral protein called HBx. But HBx is unstable on its own.
So it needs protection.
Exactly. So the virus actually hacks the host's E3 barcode scanner. It forces the human E3 ligase to scan the viral HBx protein and slap a human NEDD8 survival label onto it, specifically at K91 and K95.
Wow, it's literally using our own packing materials to protect its viral payload.
It is. And once that viral protein is stabilized by our own label, it moves into the nucleus and acts as a massive amplifier. It starts pumping out intense signaling molecules like IL-8 and YAP that trigger severe inflammation, rapidly accelerating the formation of liver tumors.
That is viral stress. What about metabolic stress? You mentioned earlier that cancer cells have broken metabolisms. Does the waste from that broken metabolism trigger the labeling machine?
It does, and in a deeply insidious way. Let's look at acute myeloid leukemia, or AML.
Okay.
The broken metabolism in these leukemia cells causes a toxic chemical byproduct called succinate to build up in massive quantities.
It's a buildup of waste.
Right, but this succinate isn't just inert waste. It is highly reactive. It physically attacks the E2 worker on the assembly line, specifically an E2 called UBC12.
Wait, the waste product is chemically attacking the conveyor belt worker?
Yes. The succinate physically phosphorylates the E2 enzyme at serine 6, structurally damaging it so it can no longer interact with the E1 worker.
So the handoff of the label completely fails.
The entire assembly line grinds to a halt. Because the garbage trucks aren't getting their VIP labels to turn on, massive amounts of dangerous, cancer-causing oncoproteins that should have been destroyed are instead allowed to pile up inside the cell, driving the leukemia forward.
Which brings us to the most tragic irony of all, oxidative stress.
Reactive oxygen species.
Right. When a patient is diagnosed with cancer, what do? We hit them with chemotherapy and radiation. But those therapies create massive oxidative stress, free radicals.
And you can guess how the cell responds to that stress. The cancer cell detects the chemical burn of the chemotherapy, perceives it as fatal stress, and instinctively upregulates the UBE2FE2 workers to churn out survival labels.
Oh, wow.
As we mentioned, this destroys the cell death triggers like NOXA. So the very therapy we use to try and kill the tumor creates the exact oxidative stress environment the tumor needs to turn on its ultimate defense mechanism.
It uses our own weapons against us. So if viral infections, toxic metabolic waste, and the oxidative burn of chemotherapy all act as the foot on the gas pedal for this rogue warehouse, how on earth do we stop it? Can we design drugs that act like a wrench thrown into the gears?
That is the bleeding edge of modern oncology right now. We call them Neddylation inhibitors. But the urgency here is massive because this rogue assembly line ruins almost every major class of cancer drug we have.
Let's walk through that resistance profile. What therapies are we actually losing to this mechanism?
We are losing tyrosine kinase inhibitors, or TKIs.
Like imatinib.
Yes, imitim used for leukemia because the overactive labeling line degrades P27, a protein that forces leukemia stem cells to stop dividing. We're also losing endocrine therapies, like tamoxifen for breast cancer, because the labeling line artificially preserves the estrogen receptor alpha that the tumor feeds on.
That's devastating.
It is. And, most devastatingly, we are losing immunotherapies. In brain cancers like glioblastoma, hyperactive neddylation degrades CMYC, which actually forces the tumor to strip away the PD-L1 immune markers that checkpoint inhibitor drugs rely on to work.
So we can't just fight the tumor. We literally have to shut down the warehouse first. Let's talk about the counterattack, starting with blocking the first worker on the line, the E1 runner.
The flagship drug in clinical trials right now is MLN-4924, also known as povonatistat. It is a brilliant piece of chemical engineering.
How does it work?
It's a small molecule inhibitor that sneaks right into the active site of the E1 worker. Once inside, it forms a covalent adduct.
Covalent, meaning an unbreakable chemical superglue.
Exactly. It literally glues the NEDD8 label directly to the E1 worker's hand. The worker can't pass it to E2. The conveyor belt freezes instantly. No labels go out.
And what happens to a cancer cell when it can't label anything?
Total gridlock. The calling garbage truck shut down. Proteins pile up by the millions. The sheer stress of this traffic jam forces the cancer cell into a state called senescence.
It just goes to sleep.
A permanent, irreversible sleep. It can never divide again.
That sounds like a silver bullet, but biology is rarely that simple. Are there caveats or drawbacks to freezing the entire warehouse?
There definitely are. While MLN-4924 forces the tumor to sleep, in certain environments, again, like glioblastoma freezing the assembly line, can accidentally trigger the tumor to display more PD-L1 on its surface.
Which tells the immune system to stand down.
Exactly. It's a double-edged sword. That is why MLN-4924 is heavily investigated not as a solo act, but in combination therapies, pairing it with immunotherapy to cover its own blind spots.
Okay, so that's gluing the first worker. What about the second worker, the E2 conveyor belt carrier?
This is where we see some incredible drug repurposing. Scientists looked at mycofungin.
Wait, mycofungin, the antifungal?
Yes, the standard FDA-approved antifungal drug used for severe yeast infections. They discovered that mycofungin physically wedges itself into the human E2 enzyme, UBE2M. It breaks the critical hydrogen bonds at GLU-107 and ASN-109 that allow the E2 worker to hold the label.
It's like greasing the worker's gloves so the label just slips right out of their hands and falls to the floor.
Precisely. And beyond repurposed drugs, researchers are pulling from nature. Compounds like arctogenin from plants and synthetics like HA9104 are being developed to specifically target the E2 workers in lung cancer, attempting to reverse that radiation resistance we talked about.
Let's move down the line to the third worker, the E3 barcode scanner. How do you stop the matchmaker?
To block the E3 scanners, you have to prevent the E2 worker from docking with them. Scientists are using DCN1 inhibitors like NSAOMHIT.
NSEMHIT.
Right. These drugs are designed to find the tiny microscopic pocket on the E3 complex where the E2 is supposed to plug in, and they just stuff it full of inert chemical bulk.
So if the scanner has a keyhole that activates it, this drug is basically jamming chewing gum into the keyhole.
That's a perfect analogy. The E2 worker bumps into the scanner, can't plug in, and the label never gets applied. Another fascinating approach uses gossypol, a natural toxin extracted from the seeds of the cotton plant.
Cottonseed?
Really? Yeah. Gossypol binds directly to the E3 complex itself, acting as a physical wedge that stops the labeling of the garbage trucks, allowing the cell's natural suicide signals to finally build up and kill the tumor.
So just to recap this section, we've learned that Neddylation causes resistance to chemo, targeted therapies, and immunotherapies. And in response, we are literally throwing gum into the gears. We've got MLN-4924 stopping E1 and repurposed drugs like mycofungin and natural compounds targeting E2 and E3.
It's a massive multi-pronged counterattack.
But thinking about the big picture here to you listening, we are using chemical superglue, antifungal grease, and toxic plant wedges to violently crash the labeling machinery. But every healthy cell in the patient's body also relies on this exact same warehouse system to survive.
Right.
How do we target just the rogue warehouse without causing catastrophic collateral damage to the patient's healthy cells?
That is the ultimate therapeutic hurdle, the defining challenge of pharmacology. The therapeutic window, the dose that kills the cancer but spares the patient, is razor thin.
Because it's the exact same machinery.
Exactly. That is why the future of this field isn't just about blowing up the whole assembly line with E1 inhibitors, but developing hyper-specific E3 inhibitors that only target the barcode scanners unique to the tumor's environment, paired with localized delivery systems that only open up inside the tumor itself.
What a phenomenal journey into the architecture of a cell. We started with a tiny molecular post-it note, NED-D8, and watched it move down a highly coordinated E1, E2, E3 assembly line.
We did.
We saw the absolute havoc a rogue manager can cause, unfolding robotic arms to fight off chemotherapy, handing out VIP passes to mutate our security guards, breaking the brakes to binge on sugar, and pulling down flags to turn invisible to the immune system.
It's terrifyingly efficient.
It is. We explored how the toxic stress of viruses, metabolic waste, and even our own keep triggers this desperate survival response. And finally, we looked at the incredible ingenuity of drugs like MLN-4924 trying to permanently freeze the gears of that rogue machine.
It truly is a masterclass in how a single microscopic modification cascades into a massive systemic disease. But I want to leave you with a thought that extends beyond just the pharmacology of waiting for the machine to break.
Oh, lay it on us.
We established that cellular stresses, like high blood glucose, massive oxidative stress from poor diet or toxins, and systemic metabolic imbalances, are the exact triggers that force this dangerous Neddylation cascade to hit overdrive in the first place.
I see where you're going. If stress is the finger pushing the button.
Exactly. It raises a provocative biological question. Could proactive lifestyle interventions habits that systematically lower baseline inflammation, balance our metabolism, and reduce systemic oxidative stress act as a natural biological brake on the Neddylation pathway?
Wow.
We focus billions of dollars on drugs designed to hit the brakes after the warehouse goes rogue, but by minimizing the environmental stress signals our cells are bathed in every day, we might potentially prevent the chaotic environment that forces the cell to hijack the labeling machine before a drug like MLN-4924 is ever needed.
I love that. Managing the warehouse environment so the rogue manager never gets the panic memo to start over-labeling in the first place.
Precisely.
So, to you listening, You now know the secret, microscopic chemical relay race that keeps cancer alive. The next time you hear a news story about a tumor mysteriously becoming resistant to a powerful new miracle drug, you'll know exactly what to ask yourself. Is the NEDD8 labeling machine working overtime in the dark? Keep questioning the mechanisms, keep diving deep, and we'll catch you next time.